In this video, we're going to talk about sugars and carbohydrates. Carbohydrates, as you may know, are the major source of energy from food in our diet. They're mostly made of the elements carbon, hydrogen, and oxygen. They're also called saccharides, which means essentially sugars. Carbohydrates like glucose are produced by plants through photosynthesis, and they are made by plants through carbon dioxide and water with energy from the sun, and oxidized in living cells to produce carbon dioxide, water, and energy. And so you can see that reaction at the bottom of the screen here. So you can see 6 molecules of carbon dioxide, 6 molecules of water being combined with energy from sunlight through photosynthesis, combining to make a glucose molecule and water. And then the reverse reaction would, of course, be respiration. So we have different types of carbohydrates. We have simple monosaccharides, and so that would be just one monomer. We have disaccharides, where we've got 2 of those. So we can see a monomer would be really simple, like honey, for example, where we've just got 1 unit. And it really can't be broken down. So kind of in the left hand, if we were to draw a line here, kind of in the left side, we've got what we're starting with. And on the right hand side, we've got if it were hydrolyzed, so broken into pieces. And a monomer, the monosaccharide, is just it. It's just 1 unit, just one ring. Milk is something that we call a disaccharide. So di meaning 2 sugars, 2 monosaccharides joined together. And so if we were to hydrolyze that, so throw in a water, we would see that it's broken into 2 separate monosaccharide molecules. And then we can have a polysaccharide. So poly meaning many, and we would put in many water molecules to hydrolyze it into many monosaccharides. And so monosaccharides can come in a variety of forms. They contain several hydroxyl or what we like to call alcohol groups attached to a carbon chain. That carbon chain can be anywhere from 3 to even as many as 8 carbon atoms long. And typically there will be an aldehyde or a ketone in that chain somewhere. If there is an aldehyde, we will call this thing an aldose. If there's a ketone, we will call it a ketose. So every other carbon atom in the chain will have an alcohol group, have a hydroxyl group, except for the carbonyl carbon. So here we've got an example of a 4-carbon chain. Every carbon present has an alcohol group except the one that's involved in the carbonyl. Our other example here, you can see the same. Alcohol or hydroxyl present in every one of them except the one that's involved in a carbonyl. Okay, so we'll call it either an aldose or a ketose. That's how we'll group them, depending on if an aldehyde or a ketone is present. The other grouping will be dependent upon how many carbons are in the backbone. So if there are 3 carbons, we'll call it a tri. If there are 5, we'll call it pent, 4 tetro, 6 hex. So this is a little bit different than we've seen previously in the class. A little bit different prefix naming in terms of our count. So let's look at some examples. Here we've got an aldehyde with 1, 2, 3 carbons. So an aldo triose. Here we've got another aldehyde with 1, 2, 3, 4 carbons present. So an aldo tetrose. Another aldehyde with 1, 2, 3, 4, 5 carbons present. and aldopentose. Here we've got a ketone with 1, 2, 3, 4, 5, 6 carbons present. So a keto, because of that ketone, hexose. Okay, so let's look at how we have these laid out. And again, we've talked about this already in the past, but how we've been seeing these monosaccharides drawn out is called the Fisher projection of a monosaccharide, where we've kind of drawn all of these carbon atoms in a T. And so here is the Fisher projection of D-glucose. And you can see that here. And D-glucose is an aldo, so here's the aldehyde. We're always going to, of course, label that as carbon number 1 because of all of the functional groups, the aldehyde ranks the most important compared to all of these alcohols, all of these hydroxyls. And we are going to look at the furthest chiral carbon from that aldehyde. So that would be carbon number 5. Carbon number 6 is not chiral because it's got 2 hydrogens attached to it, so it's not chiral. So carbon number 5 is chiral, and its hydroxyl group is to the right. So this is D-glucose. So our Fisher projection of D-glucose is shown here, squared off in purple. Our most important monosaccharides for life are typically glucose, galactose, and fructose. Glucose you're probably very familiar with. Galactose, if you're not familiar with, you will be. It is paired to form milk. And then fructose is also extremely common as well. I'm sure you've heard of high fructose corn syrup. That is the fructose part of that word. It's also extremely common in just about every fruit. So in general, the Fisher projection just represents our carbohydrates in general, where we place the aldehyde group or the most oxidized group somewhere near the top, as high towards the top as we can. And then we place all the hydrogens and hydroxyl groups at intersecting horizontal intersections across. And then we place the achiral, so that CH2OH group at the bottom. And so we can see this is an example here of L-glyceraldehyde. So you don't necessarily know the naming there, but that's okay. And here's an example of D-glyceraldehyde, so L versus D. So L, OH is on the left, D, OH is on the right. So that gives us our D and L enantiomers. L is assigned to left and D is assigned to right. So here is L-glucose, here is D-glucose. And so here is kind of those 3 important monosaccharides that we had lined up a few slides ago. Drawn out here. So D-glucose, D-galactose, and D-fructose. And you can see that glucose and galactose are both aldehydes, but fructose is a ketone. They look really similar. And you can see that the differences between glucose and galactose are really just carbons number 2, 3, and 4. Actually 2 and 4, so I'm speaking number 4 here. Here we go, number 4. So just the placement of that OH group there. Okay, D-glucose, the most common hexose that we've got in life. It's found in most fruits, veggies, corn syrup, honey. Most common, also known as dextrose. And this is what we typically use and is known as blood sugar in the body. It is the building block of sucrose and lactose and of the polysaccharides, cellulose and glycogen. And we'll see the differences between cellulose and glycogen in the next video. Galactose here is also an aldohexose, so it's also an aldehyde with 6 carbons. It is a disaccharide that is found in milk. In fact, when we bond it with glucose, it is formed in milk. It's also really important in the cell membranes of the brain and the nervous system. We do have the potential for a condition called galactosemia. And so there is an enzyme that's needed to convert D-galactose into D-glucose. And so when that's missing, galactose accumulates in the blood and tissue, which can lead to something like cataracts, mental retardation, failure to thrive, and liver disease. And so this means essentially that you can't always use it as use galactose as an energy source. D-fructose. So this is a ketohexose, so it's got a ketone. It's more, it's sweeter to the taste. In fact, it's twice as sweet as table sugar. And we often see this as high fructose corn syrup, but we use that as a sweetener. Okay, let's look at the Haworth projections of monosaccharides. And so this might be how you've seen monosaccharide sugars drawn in like a biology class. These are the more stable form than the open structure, the Fisher projection, for the 5 and 6 carbon sugars. And so when we've got a 6 carbon ring drawn out, or I'm sorry, when we've got a 6 carbon sugar drawn out in the Fisher structure, and we lay it down horizontally as such, we turn it clockwise 90 degrees. We're really going to look at creating a hemiacetal. We're going to take, in the case of glucose, we're going to take the aldehyde, and we're going to take the alcohol, the hydroxyl, from carbon number 5. And we're going to merge them to form a hemiacetal. So this is what that's going to look like. So I'm going to take this structure that's laying down, look really closely, and the first thing I'm going to do is just kind of start to curl it around. So I'm just really going to start to curl this around. And that's what you see here. It's just curled around. And then, because every single carbon-carbon bond, every single bond can spin on its own, we're going to see this bond spin. So the carbon 4-5 spin. And that's going to make it so that the CH2OH is sticking up, so carbon 6 is up, and the OH is to the right. And that's putting it in a good spot to interact with carbon number 1, the carbonyl. And in that way, we're going to form a hemiacetal. So we've got hemiacetal, remember, is an alcohol adjacent to an ether. So a hemiacetal. So when we do that, we now have what's called either alpha D-glucose or beta D-glucose. So when this alcohol here is produced, remember that alcohol used to be part of the aldehyde. So when that aldehyde is reduced into an alcohol, it can pop up or it can pop down. If it goes down, it's alpha. If it goes up, it's beta. So alpha is down, and we call these anomers. So the alpha anomer, alpha is down. So the alpha anomer is down, and the beta anomer is up. And that is backwards. I realize we always want alpha to be up. So just remember it's backwards than what we want. When it's placed in solution, it is possible for the ring structure to open and close, and we could potentially convert from alpha to beta and back. So typically at any time we could be going back and forth between alpha and beta. So usually we're about 36% alpha and about 64% beta. And the transition when it's open chain is pretty trace amounts. It's not very stable. And when it is in the open chain, it will have the aldehyde and the alcohol as if it were like a Fisher projection. So the Haworth structure of galactose is pretty similar. I'm going to go back to glucose and look really quickly. So here's the anomeric carbon. We're not going to be talking about that. I want to talk about the other. We're going to look at carbon number 4. 1, 2, 3, 4. Carbon number 4. Because that's the difference between glucose and galactose. So look at that carbon. So carbon number 4 on galactose has the alcohol up. Go back to glucose, it was down. And then also galactose is going to be forming this ring structure and still have its anomeric alpha and beta also on carbon number 1. But carbon number 4 won't change. Fructose is going to be a little bit different. Fructose had a ketone, so it can still form a hemiacetal, but it's not forming it with an aldehyde. It's forming it with a ketone. And remember that ketone was on carbon number 2. So if I can scroll back up, this is going to take a while. Let me mark this was slide 37. So if we go back here, we're going to be taking the alcohol on carbon 5 and going to carbon 2. So we're making a smaller ring. So we've actually got carbon 1. Here's carbon 2 that had originally the ketone on it. Carbon 3, carbon 4, carbon 5, carbon 6. And when it comes to alpha versus beta, we still have the same idea where the alcohol that used to be part of the ketone now can be down for alpha D-fructose or up for beta D-fructose. So remember, fructose was a ketone, carbon 1, 2, 3, 4, 5, 6. And when the alcohol on carbon 5 binds on to carbon 2, what used to be the ketone will now turn into an alcohol, and it can go in the up or down position. Oops. Okay, and that's where we'll end for this video.